Good morning all. So today I am going to discuss about the next technique, that is pour plate technique. So we can discuss about pour plate technique. In this technique, in nature actually this microbial population, it doesn't segregate themselves by species because they exist every time in a mixture of many type of cell types including bacteria, fungi, protozoa, etc. So we have the necessity to separate them into different species for the isolation and to find out which type of disease and to rule out certain types of disease. So in our laboratory, these populations can be separated into pure cultures. And these cultures contain only, what do you mean by pure culture? Pure culture means those culture containing only one type of organism. And they are suitable for our cultural study, morphological study, as well as to study the biochemical properties of each of the microorganisms. At times, it also helps in determination of viable cells. Is very crucial in many microbiological procedures. So, in the case of the microbiological investigations, there is a necessity to check whether the organism is viable or not. For all these studies, we require the organism to be in a separate pure culture. So to accomplish this, what are the different techniques we are using in our laboratory to separate? A serial dilution agar plate technique is used. And in this session, we are discussing about the pour plate. In the last session, we have discussed streak plate technique, in which the organism can be isolated into colonies in the agar media. And briefly, I am saying about this method. It involves actually a serial dilution of bacterial suspension. Serial dilution from one tube to next tube and from that tube to next tube, like that. A serial dilution of a bacterial suspension in a sterile water blanks, and it serves as diluent. So this water blank serves as a diluent to which the organism we are diluting of non-volume. Once this diluted, the suspensions are placed on a suitable nutrient media. So after the dilutions, then we are placing those dilutions or suspensions into a suitable nutrient media. And the pour plate technique, it is the procedure which is usually involved. Mainly this technique is serial dilution of the bacterial suspension. So let's check how this method comes. This is the brief picture of a serial dilution technique. And here, this is the organism, that is the mixture of colonies. So this plate contains many type of colonies including bacteria, different species, fungi, etc. And this also, there are many types of organisms, fungi and bacteria, all are present in this. So in order to separate, we are performing this pour plate technique. This is a techniques among, there are different techniques and among them pour plate technique. In this original inoculum, this is the original inoculum, and that we are adding 1 ml. So it comes now the dilution, it is, this is the diluent, that is 10. So while adding this 1 ml to this, it becomes 1:10. And again from this dilution, we are taking out 1 ml. Now this dilution becomes what? So what happens in this, one thing I forgot. Each diluent is 9 ml, okay? And we are adding 1 ml and now it becomes 10, okay? Then 1 ml we are taking it out from the first tube and adding into second tube. Now this dilution is 1:100. Then we are again taking 1 ml from this tube and adding into next tube. Now this dilution becomes 1:1000. Then we are again taking 1 ml from this tube and adding to next tube. Then it becomes 1:10,000 dilution. Then we are taking 1 ml from this tube and adding into next tube. That it becomes 1:100,000 diluent. So when the dilution is increasing, what happens? In the first dilution, the organism is very heavy. So the colony will appear like this. Then in the second dilution, what happens? The diluent increases. Dilution is more in this case. So the organism is also less. Then comparing to this each, we get separated colonies. From this it is very thick. Then after when we are plating into another, like that, now we get individual colonies can be appear in the final dilutions. Yeah, according 1:100,000. So so what is the purpose of these plates? So we have, first of all, we have made these dilutions, and each dilutions we are pouring into the plate. So this 1:10 we are taking into a plate. Like that, 1:100 into another nutrient agar plate, and then again another nutrient agar plate like this. And each of the sets we are incubating. That is the pour plate technique. And we are pipetting out like this. The sample is pipetted into sterile. And we are pipetting like this method. First of all, not directly into the nutrient agar plate. First we are taking the sample and we are pipetting into the sterile plate. Then we are applying the media. It should be not hot. If it is too hot, the organism will get die. So making it at an ambient temperature. Then we are pouring the medium above that sample. And we are just mixing inoculum by placing it on the surface and rotating, closing the lid, and we are keeping it for solidification and incubation. And later in the next day we get the colonies like this. Surface colonies are obtained as well as subsurface, means inside the agar. Two types of colonies can be seen. This is the pour plate technique in brief. So I am telling that technique in sentence. Pour plate technique requires a serial dilution of the mixed culture by means of a loop or pipette. So using the pipette or loop, because in this picture we are using pipette. So dilution is done using a loop or pipette. Then we are a molten agar, and it should be cooled to 45 degrees Celsius. And that molten agar is poured into the petri dish containing specific amount of diluted sample. Here, like this. We are adding the molten agar which is cooled to 45 degrees Celsius. Following the addition of the molten agar, then cooled molten, then cooled agar, then the cover is replaced and the plate is gently rotated. We are placing the plate on the table or platform where we have kept, and we are gently rotating in a circular motion to achieve a uniform distribution of organisms. Then this procedure is repeated for all dilutions. So we are repeating in each and every dilutions we have prepared. And this dilution should be plated in duplicate for greater accuracies. So we are not plating it into one set. We are keeping another set of plates also, duplicating for greater accuracy. And we are incubating overnight and then counted on the cubic colony counter. And later there we are using a cubic colony counter. This is the name of a colony counter. Using this we can count how many colonies appeared on that medium, either by hand counting or by electronically modified version of this instrument. We can count. If the material to be tested is an aqueous liquid, if it is an aqueous liquid,